So good evening, everyone. Welcome to another lecture on the online course of the IMQP. So tonight we have invited Claudio Berto, which is a professor at the University of Warsaw, who will talk you about changing time, changing habitats, what fossil small mammals tell us about the past. Claudio, I give you the floor.
Okay, thank you, Julie. Thank you for inviting me to this course, to this lecture. Today we are going to talk about something which maybe nobody of you knows, because it's a very narrow field of the study of prehistory and prehistoric paleontology, which is the study of the fossil small mammals. Before starting, just a little words about who I am. I am a professor of the Warsaw University Faculty of Archaeology, and I am interested in paleontology and study of paleontological sites and also study of paleoecology and paleoclimatology through the small mammals. But first of all, what is a small mammal? There is a definition. According to Andrews in 1990, he defined a large mammal as a species that weighed more than 5 kilograms in life. So that includes large rodents, lagomorphs, large carnivores, ungulates, and so on. While a small mammal is something that a mammal that weighs less than 5 kilos, such as rodents, insectivores, bats, lagomorphs, and small carnivores. Of course, there are also other definitions. For example, in zooarchaeology, they prefer to define this kind of animals like a micro-mammal, because the category of small mammals comprise a category of small prey or small game, which includes small animals hunted and collected by humans, such as small carnivores, large-sized rodents, but also fish, malacofauna, and so on. So as archaeologists, they usually prefer the use of micro-mammal term, even though both terms are correct and context dependent. For example, if I study together with a zooarchaeologist, I will use micro-mammal too, in order to be more clear. But I actually prefer the definition of small mammal because it was codified by Andrews in 1990. Of course, it is important to note that this distinction is not based on taxonomy. For example, here I like to joke with my students, telling the capybara problem, because capybara is a rodent, so it should be included in the small mammals usually on the taxonomical point of view. But actually, it weighs between 35 and 66 kilograms, and here is asking himself if he could be included in the small mammal group. So it is important to know that this definition is conventional, it's not taxonomy. We will go into it later on taxonomy. It is conventional because large mammals are treated differently from the small mammals, because they are recovered differently, they have different accumulation and so on. So why we study small mammals? There are many reasons why we study small mammals. All of them are more or less summarized in this slide. So they are reliable indicators for the constructed environment in the surrounding areas of a site. They are useful for reconstructing the past climates. They are reliable biochronological indicators. They provide useful information about the deposit formation and accumulation processes, such as they give us information about taxonomy, for example. And also they help the archaeologists to understand better the context where the past human populations lived. And of course, the most important one is they are super cute, as you can see in those photos. Going to the taxonomy, the small mammals include mostly rodents, insectivores, and bugs. But who are them? So the order of Rodentia, or rodents, is a very large order, and they can be recognized by the from the other mammals because they have these very large incisors. So they have a pair of incisors here and a pair of incisor here in the cranium, and those incisors are continuing growing. They are not provided by roots, and those together with this presence of diastema and molariform teeth, so they form the order of Rodentia. This order has a very numerous number, a very large number of species. They calculate that they are around 3,500, and they are well spread in the whole world, beside the Antarctica. Of course, some of those species, they are quite plastic on the ecological requirements. That means that they can live in forest areas, in open areas, in urban areas, no matter what, because they adapt to many kind of environments. This is the case, for example, of the rats, which are rodents that can live in forested environments, in open environments, and also they live very well in urban environments. But also many other rodents, they have strict ecological requirements. It means that they live in a particular environment and particular habitats. For example, here, the marmota, the marmot, can live only in alpine environments, or the Istrix cristata, the porcupine, can live in warmer climate and in environments which are quite from forested to dry open, let's say that. Or, for example, the squirrel that can live only in the broadleaf forest or temperate forests. Of course, other animals other rodents can be very useful for reconstructing the arrival of human population during the Neolithic, for example. This animal, Micromis minutus, is an animal that lives in crops, that loves crops, and those when we find this kind of animal, it means that probably the first farmers arrived in the region that we are studying. Other one, they are quite interesting also for the climate point of view. For example, Cricetus, they can live only in colder environment than nowadays. If you think about the Italian peninsula, those animals were present during Pleistocene, and then they disappeared. Another order of small mammals is the order Eulipotifla. Those are what they were called insectivores. They are very easy to recognize from the rodents because they have a completely different system of cranium and mandible, and also they have these kind of teeth that are made for eating insects and worms. And until a few years ago, they were discharged by the small mammal specialist. But now we realize that they are very useful for ecological reconstructions because, for example, also some of them, actually most of them, they prefer they have strict ecological requirements. Like, for example, Sorex araneus, they love to live in water edges near streams or little ponds and so on. This one, Crocidura leucodon, they love sunny sites, place with sun. Or the hedgehog, Cardinaceus europaeus, they love forested and warmer environment. Those animals, you need to know that some of them are the most smaller mammals in the world. There is one of these one species of this mammal called Suncus etruscus, which is only 1.5 centimeters long. The last order that I'm going to present about small mammals is the order of Chiroptera. Let's say the bats. The bats have peculiarity is that their peculiarity is that they can fly. They are the only mammals that they can fly. And of course, the fact that they can fly allow us to recognize them also by the postcranial elements, because the postcranial elements are very informative of the presence of the different species and presence of bats. Of course, those are also interesting from the ecological point of view, because some of them, they love to hunt near water streams. Some others, they love to nest in caves that have a particular microclimate, while others, they prefer to hunt in open environments. So as the insectivores, also the bats, we started to study them in paleoecological perspective only in the last 20 years, let's say that. So I presented the small mammals. Now I need to present also the hunters of the small mammals, because the accumulation of these animals in the archaeological and paleontological sites are not related to human, but are mostly related to nocturnal bird of prey, diurnal bird of prey, and small carnivores. We can distinguish them analyzing the fossil assemblage, because usually the nocturnal bird of prey, they leave 80 or 90% of bones that they digest. The diurnal bird of prey have an heavy digestion, while small carnivores have a strong digestion. So what they leave to the site is 20 to 40% of bones that they digest. Another peculiarity is that since they are very territorial, all of them, their hunting area is between 2 kilometers and 4 kilometers from the site where the small mammals, where they digest and they digest the small mammal. It is peculiar also to the fact that their digestion, because especially from nocturnal and diurnal bird of prey, because both of them, they are birds, so they don't have teeth, so they don't chew their prey, but they need to swallow entirely their prey. And then after the first phase of digestion, where the prey starts to be dissolved, a second phase of the digestion is like they take the nutrients of the prey in this phase, and then there is a third phase of the digestion, which is the rejection of undigested elements, which are mostly fur and bones, in form of these kind of pellets. And of course, when the pellet, the fur decompose, only the bones remains. So this is what we have when we excavate in a site. We can see the effects of the digestion in the fossil assemblage, especially the corrosion made in the stomach of this bird of prey. Which is quite easy to detect, but also in case of carnivores, we can see the breakage of the bones and some corrosion too. So analyzing this kind of modification in the fossil assemblage, we can recognize the predators. This is very important for the ecological and paleoecological reconstruction, because some predators, they prefer specific prey, so we need to take in account that. Of course, when they reject their prey, then they are decomposed, and only the bones remains, and the bones are then covered by the sediments. So how we retrieve the small mammals, since they are very small? We retrieve with washing and sorting. We wash with, of course, with water, and we sort. We wash with water and with superimposed sieve. Those sieve have usually meshes of 1 to 0.5 millimeters, but let's say 1 millimeter is enough to collect the remains of small mammals. So as you can see here, there are various examples. Usually we wash during the summer, but here we are as crazy people in Poland that we love also to wash with the snow. By the way, when you have a very good sunny times washing this sediment, then the sediment gets dry, and the result is basically that. It's basically gravel with bones and pieces of artifacts, if it is an archaeological site. And of course, inside this gravel, there are also the small mammals. So how we recover that? We recover thanks to the sorting phase. And of course, thanks to the students. Students help us a lot in the sorting phase during the fieldwork. And as you can see here, there are happy students which use tweezers and a sorting tray. When they sort, they divide the material in several boxes. As you can see here, you can see the boxes for the large mammals, the artifacts box, and this small box is the small vertebrate one. I didn't put small mammals because these include also other vertebrates. I will explain this later. And of course, when you sort, you can find many kind of small vertebrates from little teeth. This one is a tooth of a bat, bones, postcranial bones, and cranial bones. The result is basically that, is a mixing of bones and teeth, very small, of course. So how do we proceed after the collecting of this bone? These bones comes to the lab, and we proceed with the analysis. So the identification process, the first phase is to divide the small mammals from the other classes: herpetofauna, which is amphibia and reptilia, birds, and fish. We divide, of course, checking the morphology. So we are there are several specialists which can recognize what is a mammal bone from amphibian bone and bird bone and so on. Once we got the box or the bag of the small mammal, we can proceed to the identification. We do another sorting, and we basically recognize the pieces that we can determine. So we can recognize in which we can recognize the genus or species taxonomically. We can do taxonomy on that. Because we analyze the postcranial in other way, but we excluded it because teeth are the best element for recognition of the species. So once we divide the teeth from postcranial, we recognize them through microscope analysis. And of course, the species is identified through morphological analysis using comparative collections or atlases. And of course, this process must be repeated billions and billions of times, because basically our methodological approach is to record all the elements in a database. Then, of course, during the determination, we can also measure them, because some of the species can be recognized only by measuring. And of course, we do the most important thing. After we finish to do the lab work, we start to count. So we do the number of the identified specimen and the minimum number of individuals. So to counting, which is counted per layer, of course. So we ended up with several assemblages, layer by layer. Of course, after that, we proceed with the statistical analysis. So the first thing that we do is the analysis of percentages among layers. percentages of frequencies, of course, but we can proceed also to more sophisticated analysis related to statistical approach, like including descriptive, inferential, and Bayesian methods. We can use several kinds of analysis related to the presence absence or frequency tables. Of course, then we do the when we have our results, we do the interpretation, which usually is we check the other published fauna, we see which are the differences, and so on, and then all of this interpretation then needs to go, of course, to publication. Our methodological approach is based on a principle that we borrowed from geology, which is the principle of uniformitarianism. In geology, this principle explains that the processes that happen in the present can be also explain the processes that happened in the past. This can be mutuated also in paleoecology. So the biology and ecological roles of extinct organisms can often be understood by studying living organisms that are similar in form, function, or environment. That means that we can reconstruct ancient ecosystems, identify niches filled by extinct species, estimate climate condition of the past only by observing how modern species interact with their environment. Of course, this might be complicated for some of you, so I have made a very little example. For example, here we have a species called Apodemus sylvaticus. It is a mouse that nowadays lives in Europe, in the whole Europe, and also in part of the Western Africa. What we do is checking the current distribution, and also reading carefully all the information related to habitat and ecology. There is a lot of publications and a lot of books that explain species per species the habitat and ecology. After that, we gather all the information, and we in that case we end up that the current preferred habitats of these species are woodlands, woodland edges, parks, gardens, pastures, etc. And of course, using the principle of uniformitarianism, if we found a fossil Apodemus sylvaticus, in that case it is an upper molar, we can infer that the past preferred the habitats of nearby the sites might be woodlands and woodland edges. I cut it here parks and gardens because I think that in late Pleistocene such environment didn't exist yet. So of course, this is a very, very basic paleoecological inference, but actually we do in paleoecology, in paleoclimatology, we do a little more sophisticated calculation to gather all the information about the changing in habitats and environments in a specific time. So for reconstructing the habitats, we use three methods, mainly three methods. We can use also others, but these three methods are the most used. The taxonomical habitat index, the quantified ecology, and of course the most used one, which is the habitat weighting. All these three methods are related to this assumption. So first of all, we decide the type of habitats. In that case, in this case, for example, we can select a very simple one like open dry, open humid, open woodland, and so on. Then we assign a habitat coefficient to every species found in the fossil assemblage. So for example, a species which is called water vole, the Arvicola amphibius, has a wetland coefficient as 1. And then, of course, we cross-check this table with the table of the presence absent or the minimal number of individual. And what came out is the habitat percentages. And here on the right, you have an example taken from a study related to Polish caves between Late Pleistocene and Holocene. And we can see the difference in habitats. Here, for example, in this cave, we can see that the habitats tended to be more close. The forests tend to be more wide in the passage between Pleistocene and Holocene. After reconstructing the habitats, we do also the paleoclimatology inference. For doing that, we use three methods. I will not go deeper into that, but one of that, this one, the MER, use the superimposing of maps, distribution maps of species to compose a data set of climate data in matched areas. These other two use different approach. This one use species coefficient. These other ones use linear regression function to calculate the climate data. But at the end, we prefer to use all of them all together in order to compare the results. And what comes out is basically numerical results related to mean annual temperature, mean temperature of the coldest and warmest months, and mean annual precipitation. We can calculate also other parameters, for example, the aridity index. But usually in the publication you may find most of that. You may find most of this one. It ended up with more or less a description of how temperature and precipitation changes. Like, for example, here in this figure on the right, you can see how temperature rose up in Poland between the end of the Late Pleistocene and the beginning of Holocene. So it is quite, of course, there is a lot of standard deviation and so on. It's not perfect, but we can do very clear inferences on paleoclimate, especially for Late Pleistocene. Another thing that we can do with the small mammals is biostratigraphy and biochronology. So it's helping the archaeologists to understand the chronology of the site. Just a little word about chronostratigraphy. Biochronology and biostratigraphy contributes with the chronostratigraphy, which is a science that study the relation between layers and it studies mostly time, geological timing. And basically, the biochronology is the composition between biostratigraphy and geochronometry. So the biostratigraphy is the science that uses fossil assemblages to correlate and date the rock sediments layer. The assumption of the biostratigraphy and biochronology is that fossil organisms appear and disappear in specific recognizable order through geological time. What does it mean? Basically, what we do is to understand and check the first appearance and the last appearance of several taxon, including them in zones, as you can see here on the figure on the left, and the figure in the center. Here, these are the zones. And then, of course, when we check all the zones, we can also link those zones in with the absolute chronology or other proxies, like, for example, the oxygen isotope code. As here in this image, where, for example, the rodent zones were linked to these other proxies. Of course, small mammals are very useful for that because they have an evolutionary rate which is very fast, and so it allowed us to reconstruct a lot of zones, chromosomes, which are uh, they have a better definition than the zones of the large mammals, for example. Um, how do we do like that? So it's like basically in the small mammals, one of the most important thing is to check the evolution of the teeth. And in case, the most famous case is the evolution of the rooked tooth of the arvicolid, of the voles. Because basically the environments, especially in the northern hemispheres, changed from a forested one to open one, and also animals adopted. And in that case, they progressively lose their root. And paleontologists reconstructed this progression, which is not linear as this one, is actually more complicated, but this is a simplification. And they divided, studying also other species, they divided different zones. Here, for example, is a very simple example of the passage of large voles from the genus Mimomis to the genus Arvicola, and the inclusion of those mimomis in small mammal zones, which is Rushinian, Villanian, Biharian, and Thuringian. It's more or less for us, for the specialists, it's more or less for non-specialist, the same subdivision as middle Paleolithic, late Paleolithic, and so on. It's the same, but on the evolutionary trends. Okay, now that I give a very quick essay about what we can do with the small mammals, I want to show you a practical example. This is a recent example related to a site which is called Grotto della Ferrovia. In English is a railway cave. And this cave is situated in central Italy. And why we studied it? Basically, we studied because we had a context which was quite good, with a lot of small mammal sites in the northern Italy. But we wanted to understand and to describe the small mammal communities and their changes through time, especially in the last glacial maximum, which is the period that goes between 26,000 years ago to 18,000 to 16,000 years ago. It is a period which in Italy is characterized by the expansion, the last expansion of the glaciers in the Alps, and also the formation of the Great Adriatic Plain. So if you look at the position here, it is very important to recognize that Grotto della Ferrovia is one of the southern part of this area. So we wanted to go into. This cave is situated into a narrow gorge called Gola della Rossa. This gorge is created by the river Esino, this river here, and Grotto della Ferrovia open in a tributary valley of this gorge. It is situated here in the central Italy, in the region of Marche. All the data that I will show you today is published in this paper was published in 2023. So what we got was basically very few information. The first publication on 1966, a radiocarbon date of 1976 that was around 14,000 BP, and we got just a little trove of stratigraphy, no photos, no planimetry, nothing. So what we do first is to go in there, check the cave, compare the stratigraphy, and also to go to the local heritage office, which helped us and provided us the map of the cave, but also the map on where they excavated. This is a very old excavation, so they didn't use modern techniques. So it was very useful for us to find out this map, because in the labels we had these letters and we didn't know what it means. So once we established that, we studied the small mammals. And the results are indicated here in this table. Don't worry, you don't need to check all the names. But what is important is that these species here, Micatus arbolis, which is a species which nowadays doesn't live in the area, was this species was the most common in the in the assemblage. The analysis of the environment and the climate showed that there is a progression from open to closed environments, as you can see here in this scheme, and also progression towards a warmer and humid phase than in the lower part. This was nice, but actually, as you can see here from this little spoiler, we detected some problems later. So to understand this thing, it seemed like a very good sequence, but to understand the timing of these changes, we decide to date directly the fauna. Also because the peculiarity of this site was also because there were these species which is called Sichista, so in that case Sichista subtilis, which is a species which nowadays lives in a boreal area between the eastern part of Central Europe and Eastern Europe. So it's not present anymore in Italy, and also it was present mainly between the marine isotope stage 3 and the last glacial maximum. So the data that we had before of 14,000 was a little more recent, so we suspected the presence of the last glacial maximum in this assemblage. And in fact, the direct dating, direct radiocarbon dating of small mammals, which was applied for one of the first time that we applied the direct radiocarbon dating of this mammal, showed us a coherent picture on the lower layers and coherent with the last glacial maximum. But on the upper layers, you can see here on the outlier analysis that the upper layers were probably reworked because they give like inconsistent results. We used also the genetic analysis on some of the small mammals which were dated. And for example, in Microtus arbolis, this species that I described just a minute ago, we found out that the population of Grotta della Ferrovia is actually part of an extinct population of Microtus arbolis, not related anymore to the population which is present nowadays in the northern part. So this is very important. This was only preliminary. Then we discovered also that also other ancient population of Italy were part of this group. And as you can see here, this group was found also in the Balkans. It is interesting because as I show you before, this was part of the Great Adriatic Plain, so probably this area were connected during the formation of this site. Another new method that we applied was the chronological estimation through molecular data. So the results were quite astonishing because it was the first time that we applied this method, and we realized that the chronological data, the chronological estimation based on DNA, was basically similar to the radiocarbon dates. And also, it was in accordance with the polyecological and polyclimatological analysis, which show also here in the plane LGM, you show that the temperature was actually the same of the estimated temperatures for Grotta della Ferrovia derived from another model, which independent model. So it was quite similar. Of course, as you can see, the standard deviation are quite wide, but more or less we can have a picture of what changed during the LGM in this area. I think that's all for now. So thank you for your attention. Thank you for following. And now I am ready for your questions.